Hi, everyone. Welcome. Welcome to our monthly Science of All Proteins seminar. My name is Bianca Dada, and I'm the Scientific Partnerships Manager at the Good Food Institute. For those of you who are here for the first time, welcome to our community. The Good Food Institute is a nonprofit think tank that's 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based, fermentation, or cultivated meat technologies, check out our open access resources at gfi.org/science. With that, I'd like to introduce our speaker for today. Vayu is a scientist, chef, and educator who fell in love with cooking at a young age in his multicultural home in Stockholm, Sweden. He first moved to the US to work in restaurants, but the flavors, textures, and sensations of the kitchen eventually led him to scientific research. He earned his PhD in biochemistry from Harvard University, working in the lab of Emily Bauskas, where he identified and characterized strains and enzymes involved in the metabolism of prominent drugs and dietary compounds in the human gut microbiota. As a Miller Fellow at UC Berkeley, Vayu has combined his background from the kitchen and the laboratory to tackle challenges in the food system. His current research characterizes and engineers filamentous fungi to produce sustainable and delicious foods. This highly interdisciplinary work has led to new synthetic biology tools for edible fungi, discovery of a fungus domesticated for conversion of waste to food, and new dishes at Michelin-starred restaurants, including the Alchemist in Copenhagen. He will continue this work by running his own research group as an assistant professor of bioengineering at Stanford University, starting in September of 2024. In today's seminar, Dr. Hill-Maini will discuss his work bridging approaches from gastronomy, synthetic biology, and systems biology to unlock the potential of filamentous fungi for sustainable and delicious foods. Vayu, thank you for being with us here today. I'll turn the floor over to you.
Thank you so much for that wonderful introduction. And I think it's really exciting to be here, and big fan of GFI and GFI sort of advocacy and support of this space. And thank you for everybody to signing on to hear me sort of speak about how I fit into this and sort of my vision around harnessing fermentation and the power of microorganisms to create a new generation of sustainable foods that are also delicious. My name is Vayu Hill-Maini, and for the last few years, I've been a postdoctoral fellow at University of California, Berkeley with Jay Kiesling. And I'm just about to launch my own group and lab at Stanford University, just a few miles down the road from Berkeley. And today I'll share sort of the work that I've done at Berkeley and share a little bit about how it connects to my vision moving forward. But before I kind of get into the nitty-gritty and the science and sort of what I'm thinking about how we can address these global challenges, I want to share a little bit about where it all began. And my journey into this space really started in the kitchen early on in my life. I grew up in Stockholm, Sweden, in a multicultural family. And the way that I kind of connected with who I was and where I came from was through food and cooking. So it's always played a really important role in my life, in my identity, and my view of the world. And I originally thought I would be a chef. So I moved to the United States just when I turned 18 to pursue this as a career, working in restaurants, in the back of the house, working as a prep cook, a line cook, running my own kind of projects. And it was these experiences with food, really understanding what was going on, seeing what was happening, that led me to my scientific career in science, because I realized that within all this food was biology, chemistry, physics. And the science didn't only allow us to understand the food, but also to shape and modify and engineer it. And this is really what kind of kicked me off into the world of academia and scientific research. I did my PhD at Harvard University studying what happens to food and other molecules that we eat in the gut, looking at how microbes modify those molecules and impact our health. But when I came to UC Berkeley a few years ago as a Miller Fellow, I really wanted to figure out how to bring together both the gastronomy, the world of food and cooking, and my background in biochemistry and microbiology to tackle some of the most urgent challenges of our time. And the question that has really kind of engaged me and inspired me is really, how do we feed the growing population in a way that's sustainable, healthy and equitable, right? So the population is growing, we need to make food, and we need to figure out how to do that in a way that doesn't destroy the planet. Now, the current mode of food production isn't going to cut it. And I'm sure many people in the audience are very familiar with this. I'm probably preaching to the choir, but the food system is a major contributor to climate change. It's 30% of global greenhouse gas emissions. It's having a tremendously negative impact on the environment, on biodiversity, contributing to the emergence of zoonotic diseases. And yet we're going to have to produce a lot more food with a lot less land to meet the growing global demand. So there's an urgent need to kind of reimagine what we eat and how we produce that food. There's obviously many different ways to approach it. It's going to involve researchers, stakeholders, policymakers. It's a very complex challenge that's going to take all of us. And what I have really become excited about as a scientist is to look at the potential of filamentous fungi, molds and mushrooms, as one way to reimagine and redefine our food system. So why fungi? Well, fungi are not only fascinating and have all these tremendously important ecological and biological roles that contribute to the health of our planet, but fungi can also be used in a variety of ways to make sustainable foods. There's the 10,000-year-old or even more older than so technology of fermented foods, in which we grow fungi on substrates and we convert those into products with enhanced maybe nutrition, flavor, and safety. So that is really the root of using fungi in food. Now, more contemporary uses include growing fungi in bioreactors and harvesting the biomass or mycelium as a source of sustainable protein. And finally, with the advent of CRISPR-Cas9 and gene editing technology, we can actually program fungi to produce useful molecules, proteins, and other ingredients in more sustainable ways. For example, using fungi to produce large quantities of dairy proteins instead of using cows. So fungi really have a tremendous potential, I think, to play a role in our food system and meeting these demands. And the way that I see it is really kind of from kind of a multidisciplinary lens and really thinking about all that fungi have to offer. So the last few years, my work has really kind of thought about how can we study fungi? How can we discover fundamental biology and processes that help us tackle global challenges? So the realm of learning and discovery. How do we then bring in contemporary genetic and tools and technologies to push the capabilities of fungi to meet our demands and expand beyond naturally existing biodiversity? And finally, how do we then put it all together, what we're learning in the lab, the genetic and biochemical insight, to make an impact in the world, actually translating our knowledge into tangible solutions? And here's where I've worked with chefs. So, you can see here that this work has spanned kind of a lot of different focus areas, but really kind of along the line of trying to harness fungi to make food. And I won't talk too much about our efforts in synthetic biology and engineering today. You can read the paper that came out earlier this year. But what I really want to talk is how we can kind of bridge discovery, learning, with working with chefs to make an impact. This is kind of the story that I'll tell. And the work will be mainly focused on a paper that was just accepted in Nature Microbiology that will be published over the coming weeks or months. And this is, again, learning from nature, discovering kind of solutions that might help us address pressing challenges in our food system. So the challenge that we focus on here is the problem of food waste. In industrialized countries such as the US, over 30% of food is wasted. This accounts for over half of greenhouse gas emissions of the food system. And you can imagine that by wasting food, we're not only having a detrimental environmental impact, but also this presents issues for food security. If we were able to actually make more use of the resources that we have, we would be better positioned to feed our population and address challenges and inequities in food access. So minimizing food waste is critical for improving the sustainability and sort of the security of the food system. And when I say food waste, it's not just eggshells and kale stems in your trash can, food waste really occurs on a massive industrial scale. What happens to all the soybeans that didn't make it into the soy milk? That's a waste product that is edible. We could make use of it, but we don't. So those type of waste sources are called byproducts. They're basically large-scale industrial byproducts that are coming out of our food and agricultural system. Now, the idea here is that if we could take some of those byproducts and we could convert them back into human food, we not only solve the sort of waste issue, but we also solve, the food issue. And fungal fermentation is a particularly promising approach to take byproducts and convert them back into human food. We know that through fungal fermentation, we can change the sensory attributes of these byproducts. We can enhance the nutritional value. It becomes more palatable to us as humans. We also know on more a global kind of impact scale that this has massive food security, sustainability benefits. We can save a lot of CO2 that would otherwise be resulting from landfills. So this is the opportunity. This is the direction. A lot of people have been looking at it, including chefs in various contexts. But to me, kind of addressing this, I was really wondering, where do we start? There's so many fungi, there's so many foods we could make, and there's so many byproducts. So finding the right combination to really hit the spot on sustainability and flavor is the challenge. So what I decided to do is actually to learn from an existing practice that has figured this out. And this is the production of oncom, a traditional meat substitute produced and consumed on the island of Java in Indonesia, particularly in the western part of Java. And this is really intriguing because it is an example of fungal upcycling for creating food. There's two types of oncom. Black oncom is made from peanut oil residue, and that's soaked and fermented, producing black oncom. And you have red oncom, which is made from the byproduct of soy milk production. All the cellulose-rich fibrous soy biomass converted into this product. And you can see they have really unique appearances. They're sort of similar maybe to tempeh, maybe a more familiar food for many people, a block that's sliced and added to soups and stews. And I just want to highlight the incredible versatility that these foods have, being used as kind of a center of the plate protein or stir-fried with vegetables in different grain dishes, as a snack. This is something that clearly is delicious and used in a variety of ways. And I think if we can learn from these processes and study and understand what's going on, we might actually inspire solutions in other cultural contexts and address some of these challenges that we're facing. So what we wanted to do is actually to understand, how does oncom work? We know sort of that A goes to B, but what has actually happened inside? And this is the story I'll tell today. And for this, we teamed up with my collaborator in Indonesia, Professor Hani Wijaya, as well as an international team, including Pablo Cruz Morales in Denmark at DTU, Jose Manuel Villalobos at UC Berkeley, Nabila Rodriguez Valeron from the Basque Culinary Center, chefs at Alchemist Restaurant in Denmark, and the Joint Genome Institute in Berkeley. So really an international team that brought a lot of different perspectives and skills to tackling this question. And this really wouldn't have been possible without all these amazing contributions and insights. So the first thing that we did is that we went to Indonesia and we collected samples. So oncom is produced on a very small scale. It's still sort of practiced as a cottage industry. We went to different producers and we collected black and red oncom samples from these producers. And then we used what's called metagenomic sequencing, trying to understand what the microbes were that were involved. And I want to say that this is not done with sterile biosafety cabinets or autoclaves. It's done in a very humid environment where a lot of microbes could grow and thrive. And so we were wondering at first, what microorganisms are helping shape this process? And you can see here some of the samples we collected, different producers. There's sort of a uniform size and shape. For red oncom, you can see that they're sort of red covered in this orange covering. And what I want to show you now is the results of our metagenome analysis. You'll see on the x-axis, different sort of either bacteria or eukaryotes. On the y-axis, you'll see either red or black oncom, where these were found. And it's going to be shown as a heat map, where more intense red color shows more presence in these samples. And you can see that between samples and producers, there's some variability. But what really stood out to us is the patterns found in red oncom, which was that they were basically dominated by a single fungus. called assigned to the Neurospora genus. And because of this very unique dominance and sort of low variability among red ancham samples, we decided to dig into this a little bit deeper. And upon reading the literature and sort of cross-referencing our sequencing data with some genome sequencing that we had, we unveiled that Neurospora intermedia is probably the species of fungus that is involved and playing the major role in red ancham production. So Neurospora intermedia had been isolated from ancham for decades. People have taken and cultured ancham and seen, oh, look, this is growing. But our results really reveal that this is probably playing a dominant major role. And the fact that it's the only organism we can detect in some samples suggests it's probably sufficient to take soy milk waste and make the food from it. So now that we have the organism, we wanted to figure out a little bit about what's going on. You know, what allows this organism to thrive on the waste? There must be something here about its growth and ability to consume this material to convert it so rapidly. So one thing we noticed is that when we analyzed the fermentation process before and after, we found that the fungus released sugars as a result of the fermentation. So whereas in the starting material, all of the sugars are bound up in complex biopolymers such as pectin and cellulose, after the fermentation, we saw production of arabinose, galactose, and glucose, suggesting to us, based on our knowledge of fungi, that probably what the fungus was doing was maybe secreting carbohydrate active enzymes that are chopping up carbohydrates or polymers and then using the sugars to support its growth. And this is a common way that fungi break down plant biomass. So with this kind of biochemical data, we wanted to try to identify what enzymes might be involved. Were there actually carbohydrate active enzymes? What were they breaking down? And in understanding this, trying to understand what the fungus was actually eating. So here we did what's called RNA sequencing or transcriptomics. We grew the fungus on the substrate, the soy milk waste or okara, and then we looked for genes that were highly expressed during the growth. And you'll see here on the y-axis how much that expression was induced. And the color of the dots is that the green suggests that it was different, compared to a control. The pink suggests that it's a carbohydrate active enzymes. And we're going to look above a certain threshold where we think that's the most significant. And you can see a lot of things are changing. Obviously, we're growing it. A lot of genes are being expressed. But above this threshold, there was a few key genes that stood out, including a bunch of predicted carbohydrate active enzymes. So our hypothesis that probably the fungus is producing carbohydrate active enzymes to degrade the soy biomass was, seemed to be true. But when we looked into it, we realized this doesn't actually mean much at all. It was hard to make sense of it. We got a list that you can see here of different gene names, but what is the fungus actually eating? And what are the enzymes actually doing inside the biomass? I want to highlight here that the okara or soy pulp is a complex product composed of the soy cell wall, pectin, cellulose, polysaccharide, oligosaccharide. So, we're looking at a substrate that's kind of messy. And what we decided to do is to actually say, let's break down that substrate and look at all the different components that we have inside the complex plant, avicell or cellulose, different polysaccharides, rhamnogalacturonnan, stachyose, raffinose, sort of components that we can actually purify. And let's repeat the same RNA sequencing experiments and compare the expression of genes in the very complicated plant setting with purified carbon sources. And by comparing the gene expression, we might be able to understand what these enzymes are doing and what they're actually involved in. And this turned out to be a very fruitful effort. You can see here a heat map that shows really nice kind of overlaps or lack of overlaps between the complex okara on the left and other carbon sources. But what I want to focus on for the purposes of today is this amazingly clear overlap between the gene expression on okara or soy byproduct and pure cellulose. So there is a collection of genes here that clearly seem to be involved in cellulose degradation, suggesting that one of the important processes that's going on is probably the fungus is degrading and eating cellulose. So to confirm this genetic insight, we turned to looking at the biochemistry. And when we looked for enzymes that are being secreted by the fungus, we found that it's cellulose degrading enzymes. When we grow it on sugar, it doesn't have to degrade any cellulose, so it's happy. It doesn't produce any cellulose degrading enzymes. When we grow it on avicell or cellulose, we see that those enzymes are being secreted. And the same thing is true when we grow it on soy pulp or okada. So we have kind of moved here from sequencing to actually confirming a key biochemical activity, suggesting that the fungus is consuming and breaking down cellulose, which is supporting its growth in making this fermented food. So I think this to me was kind of a key insight, because humans cannot degrade cellulose, but the fungus can. And so we have sort of turned to the fungus to maybe eat it, make more of itself, and then transform the substrate. And that raised a really interesting question of how do we get here? How did we start working with this fungus as humans, and what is its evolutionary history? And this must give us a sense into how we arrived at this really interesting fermentation process. So what we did here is really we teamed up with the Joint Genome Institute, and we sequenced a bunch of different strains of Neurospora intermedia. You can think of Neurospora intermedia as the equivalent, you can say Homo sapiens, but Homo sapiens humans, we have different individuals. And the same thing is true in fungi. We have different strains or different individuals of Neurospora intermedia. And we took upon ourselves to sequence these and then to compare all those genomes, their DNA sequences, to try to understand how they're related to one another. And I think this presented one of the most exciting findings of our research that we did. So what I'm going to show you here is a tree where different Neurospora strains are shown as the tip of the branches, and how closely related the branches are to one another sort of indicates their relatedness, much like a family tree. And on the bottom, you're going to see Neurospora species, not Neurospora intermedia, just other Neurospora species that we use sort of as a reference. They're not relevant to our study and not relevant to Ancham, but they help us kind of ground and root the tree. And then the two clades that you see here are very intriguing, because there's two groups of Neurospora intermedia. One group on the top that's gray, and one group on the lower group that is orange. So when we look for the Ancham strains on this group, we see that they're all closely related to one another, as we might expect. You can see here in bold, in orange, you have Neurospora intermedia from Ancham, from Indonesia. What was really interesting is that these strains are closely related to strains that are found on other human byproducts, plant-based cellulose-rich byproducts. Sugarcane fiber from Taiwan, for example, the main byproduct of sugarcane production. When humans make sugar, we press the sugarcane juice, and we have this fibrous biomass left over. And corn cobs, the leftover of humans eating corn and throwing out the cellulose-rich corn cobs. Now, you can see that essentially Neurospora intermedia exists as a byproduct associated group that is distinct from the wild relatives that are associated with burned vegetation. So Neurospora intermedia, the sort of wild strains we think of as growing on after a forest or a grass fire. That's where we find them. But you can see an association of a distinct group that seems to have evolved towards growing on byproducts, human, cellulose rich of human food production. And so this kind of suggested as maybe a kind of an interesting domestication or association with a particular lifestyle of human byproducts. And to dig deeper into this, we wanted to analyze and sort of understand whether there were any interesting genetic or biochemical differences between these two groups. We see that, yes, they are genetically different, but what actually is going on a more mechanistic level? So we looked at all the mutations across the whole genome, looking at so-called SNPs or single nucleotide polymorphisms. And we found that indeed these two groups segregate based on their mutational profile. That if you look at how many kind of nucleotide changes we have, you see that they're different. And then when we cross-reference across the genome, we cross-reference these changes to the actual transcriptome or the gene expression data we had from growing on waste. We found something really interesting, that one of the most highly expressed cellulose degrading enzymes or cellulases that was highly expressed on okada or soy pulp was highly mutated in the burned vegetation or wild strains relative to the byproduct group, suggesting that maybe there could be some important or interesting differences in cellulose degradation, which we know, again, is important for growing on this waste. And when we look at the biochemistry, in fact, that's what we see, that the byproduct associated strains, the strains that we think have evolved together with human byproducts, have much higher, about 4 times higher, secreted cellulose degrading ability or cellulase activity relative to the burn strains. And while we cannot conclude that these results are directly from this enzyme, it does suggest that there's a broader kind of change that might be relevant to the growth on waste and byproducts. And this would not be completely wild because, or strange, because we know from other fungi through human domestication that we have changed genes and enzymes that are really important for the fermentation characteristics that we care about. And I'm just really kind of intrigued here to think about what this might mean. And I also just want to share that these results were really enabled by the amazing genomic skills of Pablo Cruz Morales and his group in Denmark. So, let's hear that Neurospora grows on byproducts. We know it can degrade cellulose really well. We wanted to next kind of take a step back and try to understand whether these findings could be relevant in a broader context. Soy pulp is not the only waste produced in massive quantities from the food industry. There's a bunch of others that are being produced. And we wanted to see, knowing about Neurospora and its ecological and evolutionary sort of context, could it grow and break down other waste products? So here's where I partnered up with the food industry and restaurant partners. I assembled a collection of 30 different industrial grade byproducts that are coming out of plant-based milk production and so forth. And then we grew Neurospora intermedia and assessed its ability to grow in solid state without any additives at all. And this was done by my talented undergraduate, Alexander Rosales. And we kind of called this Project Wasteland because we just ended up in the lab having all these different wastes and we were just assessing growth. And I think this is a really exciting finding here where Neurospora intermedia has the ability to grow across a range of different byproducts from plant-based milk production, including oat milk, one of the major plant-based milks on the market. The byproducts of that are very, very good for Neurospora to grow on. Fruit and vegetable pomace, right? You can see just the variety here in its growth. In just 36 hours, it's completely penetrating and consuming these waste streams. From grain and agricultural processing, almond hulls, rice hulls, from brewing industry, from the oilseed production, and other byproducts that are really relevant. So Neurospora intermedia has a really impressive substrate scope. It rapidly degrades in just 36 to 48 hours, all these byproducts, changing what they look like, and maybe other attributes as well. So I think these results here really establish kind of a relevance to maybe the broader challenge of waste upcycling. Maybe Neurospora intermedia could be deployed in various cultural contexts as a solution for dealing with waste. And one of the things I think that we wanted to figure out to see whether this is a real possibility is it safe? Is it nutritious? And do humans actually like it? So far I've shown you all these photos, but what does it mean if we don't actually sort of perceive it in a positive way? So the first thing we did is we looked for potential toxins. So many fungi, including the fungi that are sometimes used in fermentation, have the genetic capacity to make mycotoxins, toxins that are detrimental to human health. Now, many of these fungi do not make mycotoxins in the traditional context where they're being grown. Let's say they're growing on rice, that's how we use them in fermentation. They're not going to make those toxins, but the capacity to produce those toxins could present a danger if we want to grow it on a different substrate, which we didn't select and evolve it for. So what we did here is we searched the genome for natural products and secondary metabolites, including toxins. The key takeaway is that unlike many other fungi, Aspergillus oryzae, Aspergillus sorghum, penicillium strains, fusarium strains that are used for food, Neurospora intermedia does not have the capacity to produce any known mycotoxins, and it also doesn't produce many natural products at all, which is kind of interesting. And this sort of supports the historical evidence that's out there in the literature that Neurospora might be safe, and recent approval for Neurospora as mycoprotein by, I think, the USDA. So, this is sort of consistent with Neurospora as being a fungus that's safe to consume from a toxin perspective. The other thing we want to ask is it nutritious, right? So maybe some of these plant-based milks or other byproducts are not so nutritious in their own. They're full of cellulose and fiber. We wanted to ask whether Neurospora could elevate the nutritional value. And in very preliminary data that we're still sort of exploring, we see a strong, consistent kind of increase in protein when the fungus is growing, assimilating that nitrogen and producing more of its own mycelium and protein, suggesting that there might be a nutritional benefit to growing. And I'm not showing those data here today, but we also see the production of various bioactive metabolites and vitamins by Neurospora. So, we think that there could be a nutritional benefit to cultivating it on these byproducts. But I think for me, as a former chef, really importantly, we have to think about whether it's palatable, right? Some foods are deeply cultural and do not have appeal outside the context where they're usually produced and consumed. I think about surströmming, which is a very polarizing fermented fish product from Sweden, where I grew up. I don't think it's the biggest world-changing kind of technology, because it is frankly very difficult for many people to consume. So what about Neurospora? How do people perceive it? What is its impact on both the sensory, aroma profile, but also the kind of human dimension of the waste? So one thing we notice is that there's a complete change in the volatile aroma composition. So for example, when we look at what happens to the soy milk waste, the beany cellulose-rich biomass of soy milk waste, what happens? Well, for example, we see the decrease in hexanol, a major off flavor, a grassy, waxy, and beany aroma that's very strongly linked to legumes. And it's something that we often want to get rid of if we want to make protein isolates from pea or soy. And we can see that in the fermentation process, Neurospora, basically, it seems to disappear. And when we smell it, we don't smell anymore of the green, grassy kind of legume flavor. The aroma has changed completely. And we see that in a broader way in data that I'm not showing here today. The other thing we did is actually brought this Neurospora intermedia, and we made some foods from waste, and we tested it with Danish consumers in a collaboration with alchemists. And we asked them how to describe the attributes of what they were eating, and also whether they liked it or didn't like it on a qualitative scale. And this is 61 Danish consumers. I would say Danish people are kind of picky, so I cannot feel like I trust the data. But of course, it's a small population. It's just a starting point. But what we found is that basically people assigned positive attributes to the food. So rather than off-putting, astringent, and disgusting, we see attributes such as nutty, mushroomy, fermented, brown, earthy, attributes that are not thought of as negative. And then when we ask them to like it on a scale from 0 to 9, where basically this is the most disgusting thing you've ever had, or this is the best thing you've ever had, anything above a 5 is showed a liking towards the product that, I perceive this positively. And this is a product that's not seasoned or modified in any way. It's just the raw thing that's fried in the pan. And people were open to it. They have showed a liking towards it, consistently rated about a 6 or above, which I think is really exciting that, this has maybe some sensory appeal beyond the traditional cultural context where we consume and produce it. Suggesting that Neurospora could kind of move is, be an interesting solution as we think about food waste and upcycling into the future. So, the big takeaway that we have here really for me is to kind of look at this as a continuum where, through culturing and consistent kind of growth of Neurospora, we're able to probably domesticate a fungus that traditionally grows in the wild, and we can turn it into something that grows readily on byproducts. When we started making byproducts, our hypothesis is that it created a new niche for Neurospora to grow on. So it started growing on byproducts. And one of those byproducts might have been soy milk waste. And that is maybe then what led us to make this delicious food. And we can see some genetic changes and enhanced capacity for degrading cellulose. And I think moving forward, if we look at this continuum, maybe this could be a really promising solution for food security and sustainability. The ease of cultivating in solid state, there's a low technological barrier of growing fungi in solid state that compares to large-scale precision fermentation in steel tanks. I think it makes it accessible as a technology. I think it also is culturally adaptable because byproducts and food preferences, it seems to be flexible to that. And, really exciting about, the possibilities moving forward. And I think that to make that happen, you know, one of my goals has been to really try to bridge this discovery and learning, you know, with actual impact, and think about how we can take our understanding of the biochemistry, the genetics, the safety, the flavor, and try to translate that into actually making food, right? So often in academia, you know, we're doing really cool science and we think and we talk about it and we present it. But I think it's especially important in food that it doesn't stay just as academic publications. And we actually try to, you know, have engage with stakeholders to try to bridge our knowledge with expertise and production on the ground. So now I'm going to talk a little bit about some work that I've done in this space. And here's where we initially teamed up with a restaurant called Alchemist, you know, ranked as one of the world's best restaurants, a very creative place, very forward thinking, very science forward, and very sustainability oriented. So what we did is I brought Neuraspora intermedia into the kitchen. You know, it's non-GMO, it's safe to eat. And we started building capacity among this amazing kind of production team, setting up a little bit of a lab, you know, installing a bioreactor, and then really getting to work. And it's a beautiful kind of synergy where, you know, we're providing the insight around the protocols and the processes, but chefs really guide us as to where this can go. What are the flavor profiles? What are the textures? What are promising applications? And I don't want to go into deep detail with everything here, but we have so many exciting prototypes here for mycelium-based meats, center of the plate proteins, new beverages, fermented grains and legumes, and even plant-based cheese. And what I want to highlight is a little bit how kind of the gastronomy approach led us to innovate in a new way, harnessing Neuraspora intermedia's enzymes to completely transform plant biomass into new foods. So one thing we noticed is that when we grew Neuraspora intermedia on rice, we not only completely transformed the color of the rice into this orange-looking cake, but the flavor, creating a sweet rice that had notes of flowers, fruits, ripe fruits, bee pollen. You know, completely the sensory profile had changed. And, you know, we noticed that sugars were probably being produced because we could taste the sweetness. And whereas in our discovery study, we had analyzed using LCMS and metabolomics to discover sugar changes, here our mouths were telling us the same stories. And it led us to think that probably carbohydrate active enzymes were being produced. Fungus was growing on plant polymer, secreting enzymes to degrade those polymers, releasing sugars that were then sweet. And we found then a way to actually harness these enzymes to completely hydrolyze and modify the texture and flavor of the rice. So what we did here is that we were able to grow the fungus on the rice. Then we have a little cake and we add a little bit more rice and water, and then we heat that up. And those enzymes that were produced were sort of hydrolyzing everything, creating a plant-based beverage that's naturally sweetened through the action of fungal enzymes. Very similar to what you might know as amasake in traditional Japanese fermentation. And so again, here you're finding ways based on the biochemical understanding to unlock new gastronomic applications, kind of creating a plant-based, you know, beverage that has water, fungus, and rice as its only ingredients. And what we found is, in fact, that compared to other fungi that are used for starch to sugar conversion, Neuraspora intermedia surprisingly is as good or better. For example, Aspergillus oryzae is used in East Asia to make sake and breaking down starch to make sugar. Neuraspora intermedia is perfectly suited for this purpose. And we discovered that through the biochemistry in collaboration with chefs. And that now lays the basis for a new dish on the Alchemist menu, where we harness the enzymes to create a naturally sweetened dessert. We create a rice pudding, kind of like an agar. We grow the fungus on top of the rice pudding, creating this beautiful kind of orange appearance, but also modifying the sweetness because the fungi secretes enzymes. It becomes a lot more sweet, and the aroma profile, those ripe fruit, kind of bee pollen aromas. And it was really incredible for me, you know, as a scientist, having worked as a chef, to have the experience, you know, during the Miller Fellowship, to see these results materialize and manifested in an actual food. Going to Alchemist with Professor Jay Kiesling, who was my, you know, postdoc host, with Chef Rasmus Munk, and engaging and tasting this dish in the restaurant. You know, really showcasing how a biochemical understanding and a connection to gastronomy can help us accelerate this innovation and get it into the hands of people. Now, what I'm really excited about is actually, okay, we've learned here about Neuraspora intermedia, we can do different things with it. But what I want to do is to circle back to those findings that it grows on different waste products or byproducts. How do we convert those byproducts into a range of new culinary experiences and foods out there in the world? And so that's where, in a very early collaboration, but a really exciting collaboration with Blue Hill at Stone Barns, as well as Stone Barns Center for Agriculture and Food in New York City, outside of New York. Dan Barber is the chef at Blue Hill, one of the leading voices in sustainable gastronomy and sustainable food production, and how we can use gastronomy to shape that. We have really teamed up here in an exciting collaboration to take all those findings about growth on waste and turn it into new foods. So the first thing we've done is we've built capacity. So we have set up a full-scale kind of food-grade microbiology and fungal fermentation lab on site at Blue Hill at Stone Barns with the support of Stone Barns and Blue Hill. We're able now to cultivate and isolate and study fungi here, a really unique resource. So again, for me, it's about scaling through capacity building and helping educate and empower these stakeholders with these solutions so we can implement them in these different contexts. You can see here is a chef team, running workshops, talking about fungi, the power of fermentation, what are the solutions, what are the promises, what are the challenges, getting their perspective on the production bottlenecks that we might run into, just really engaging across these disciplines, and then getting to work prototyping. Massive, production of byproducts from agriculture and food at Blue Hill and Stone Barns, growing it, testing it, what are promising, again, production modes, tasting it. I think this has been one of the most rewarding parts of the experience the last few years. It's just really engaging with the chefs, bringing my knowledge, but also learning so much in the process. You can see here a range of different prototypes and applications that have emerged. And I'm not really ready to share, the details of this yet, but it's to say that this is, I think, has a lot of promise and potential. And I would invite everybody here on the call, who might be interested, to think about how we might work together to build these capacities in different contexts. And the vision here, what we're moving towards, is a new generation of nutrient-dense, climate-smart, and accessible foods that happen to be made from byproducts. We're not trying to say, oh, this is a food that you must eat because it's sustainable and it's made from byproducts. The idea is that we're making delicious stuff. And guess what? It also helps us create a more circular food system and a more secure food system. And if you look at something that I made here, recently as a result of one of our prototypes, I think this is just, where it can go. Maybe your frozen patties that you're buying in the store will be fungally fermented byproducts that deliver on nutrition and flavor, which makes us want to buy it again. So that kind of showcases a little bit, the vision that I've had and the work that I've been doing. It's really discovery. There's so much to uncover in the fungal kingdom, to uncover about fungi and food, and then how we then take that to build capacity among stakeholders, working with, learning from chefs to bring these solutions in the world. And I think this is a new, exciting way of working. And I'm excited to continue that work. And I think more people should be really engaging, not only with the technological aspects of food, but also the cultural and sensory dimensions, because I think that is a critical piece in adoption and, changing food systems on a larger scale. Now, what I didn't talk about so much today is actually our efforts in synthetic biology. Saying that, okay, we have these domesticated fungi, we've worked with them for hundreds of years. How do we push their capabilities using contemporary gene editing tools to program what they do for us? Our needs at this moment are a little bit different from our past. Our capacities are different, and we're able to grow fungi and use fungi in different ways. So I think synthetic biology has a big role to play here. And one of the things that we did in the paper that is published, and you might want to look up after this, is develop a synthetic biology toolkit for an edible fungus to program and completely alter its nutritional value and sensory appeal for meat substitutes. You can see here, for example, how we engineered heme production in the edible biomass to create a red, what looks and tastes like red meat without any additives or any other modifications. And I think I'm excited to kind of lead the frontier and continue some of this work moving forward. Now, this is really work that was done at Berkeley, to the support of the Miller Fellowship and my collaborations and the support of Jay Kiesling. And I'm really excited to be talking to you right at this moment because I'm about to start my own group at Stanford University, fully focusing on harnessing fungi for food and sustainability. our website is live if you want to check it out. We'll be affiliated. I'll be in the Department of Bioengineering, but I'll have strong connection to Interdisciplinary Biology Institute, BioX, and the School of Sustainability. And I'm really excited about Stanford because I think at this moment in time, they're really embracing sustainability and putting that at the front and center. How do we take all these technologies and insights as academics, putting it towards planetary health and addressing global challenges? And also, I think, embracing more and more food as being part of that. And so I can see this, my lab, but also this area growing at Stanford. And I think it's going to be a really exciting place in the next few years, in the decade, to expand and build around food and sustainability in this way. And my lab will really build on the efforts you've seen. You've seen how we center on fungi. They have so much potential and promise. We bring in approaches from biochemistry, from synthetic biology and gastronomy to study, understand these organisms, right? To understand what's going on, how do they work, to engineer these organisms, and then to translate our insights into solutions and impact on the ground. Whether that be new companies, new technologies, new knowledge for chefs, for producers, for academics. I really want to kind of continue working across this spectrum. And while we will focus very strongly on food, there is so much kind of more to fungi and the potential for planetary health. Some exciting new directions and projects that we'll be pursuing includes understanding fungal domestication. How does this happen? What are the steps that happen in the process from going from wild to domesticated fungi for food production? How can we understand the genomic, the genetic, and biochemical details of those steps to then program fungi and domesticate them for the future? What if we could rationally take fungi and domesticate them for certain purposes in the way that our ancestors have done through continuous cultivation and growth? Our ancestors and the cultures that came before us really are bioengineers and scientists, and we're just continuing that legacy. And I'm excited to see how we can continue it with new technologies and insight. I'm really excited about mushrooms. I talked today about molds, Neurospora intermedia as one of them. Mushrooms are another type of filamentous fungus with incredible potential for medicine, for food, for materials. How do we then understand mushroom biochemistry and function? How do we understand the form? And then how do we tap into that to make new materials, new chemicals, enzymes in sustainable ways? And I think mushrooms are really cool because they can grow on lignin and wood. And there's a whole kind of untapped feedstock there that we can grow them on. We're also looking at fungi for agriculture. Fungi have a tremendous impact on agriculture. Yes, they are pathogens, they are devastating, but there's also fungi that help control other pathogens and help boost plant growth. So we have some grants already actually for studying how these interactions happen. How do fungi interact with plants, with other bacteria in the soil? And then how do we actually try to engineer and modify those interactions to benefit agriculture in the traditional way? And then finally, looking at upcycling, the work with Neurospora, continuing to build the capacity among stakeholders, leverage these gastronomy forward approaches, but also to engineer the process of upcycling. Can we start using genetics to say, we want more of this vitamin, we want more of this flavor, we want more of this texture? I think there's a lot of room there for genetics and synthetic biology to come in. And one unique aspect of my future lab at Stanford, which is one of the reasons I really wanted to come to Stanford, is we're going to be having a macrobiology lab and next door a full-scale R&D kitchen, inspired by my times in these Michelin-starred restaurants and working in these different gastronomy contexts. You can imagine here the photos are representing learning and studying what's going on, but then actually prototyping and engaging stakeholders. So that will mean that we can invite people from the outside world in, but we can also be taking our technologies and insight, to the world outside. And I'm really excited to get this R&D kitchen, up and running. And right now we're recruiting PhD students, postdocs, and chefs to join our group. And we're looking to, we've already raised some funds, and we're raising some funds to launch a chef in residence program, exposing chefs to, CRISPR and fermentation, and try to find kind of synergies for sustainable food innovations. And with that, I just want to acknowledge all the people who have been part of this journey, from my PhD onward, including my collaborators who made this possible. A particular shout out to the chefs who were crazy enough to engage with somebody like me. And I'm excited for continuing to build these collaborations across cultures, across disciplines to advance this field. And I also want to say that, while we have started to work with these, Michelin-star restaurants and these very exclusive contexts, I think this is just a launching pad for bringing this as an accessible kind of solution for people in the world. So I want to highlight that, the goal isn't to be making more Michelin-star meals, is to be using these collaborations and resources to create solutions for the world. And with that, I want to open it up for questions. You can contact me at my Stanford email, follow our Instagram, Twitter, or X. And yeah, thank you so much to everybody who was here to listen to this talk and, this research that I've been doing. And I'm excited to hear your questions and figure out what might be in store moving forward. Thank you again.
Thank you, Vayu, for that really amazing talk. It's really inspiring to hear how you were able to really bridge this fundamental research with actually getting things out there and hearing how consumers react to it. I think that's quite rare to see in one scope of research. So
very full circle for me too. It's almost like I'm just that old, like the photo I showed in the beginning where I fell in love with cooking. It's like I'm cooking, but just in a different way, in different context. But it's really cool to circle back,
Yeah, it's amazing to see that. We have a lot of questions in the chat. I'm going to start with one that builds off of that last point that you made about accessibility. So can you speak a little bit to what scaling looks like for this, both in terms of cost and potential barriers, and then also kind of building on this idea of working with these Michelin-star chefs? How are you thinking about creating, making this more accessible just to the global masses and things like that?
Yeah. So I think you can think of scaling in a few different ways, right? So a lot of the times how we think about scaling, at least in the kind of the Bay Area and Silicon Valley and food tech, is like, okay, we got to get a lot of steel in the ground, make a giant factory. And then from there, we produce at scale, so to speak, and we are able to kind of deliver these products to people. I think another way that I've become really intrigued with around scaling is scaling through in a distributed fashion, right? By building capacity in different local contexts for people to implement these solutions in ways that are culturally relevant and make sense for them. So I really think about this capacity building, right? Of bringing people in, teaching people how to grow fungi in safe ways and meaningful ways, and saying, okay, what is the byproduct? What is the challenge in that context? And how can we kind of work together to find solutions there? And I think that for me is a really meaningful way moving forward. We'll have the R&D kitchen. I want to host people, I've applied for a grant from the USDA. We'll see how that works. To really host workshops around fungal fermentation and sustainability, right? And make this accessible. And I think that as far as impact and scale, I think that's how we ultimately can have an impact, because there's buy-in and there's cultural relevance kind of in these ways. Now, if you look at the numbers, what I've seen in fungal fermentation, of course, there's challenges. You think about solid state fermentation. We're going to take the substrate and we're going to grow the fungus on it, and bam, we have our food. Now that's very accessible, and the numbers suggest that actually delivers on sustainability. There's challenges in logistics and transport and spoilage. And so I think there's definitely some key production bottlenecks and sort of production methods. I was very inspired by my time in Japan 1 year ago, realizing that solid state fermentation has been figured out at massive scales. Soy sauce, miso production, we are growing fungi on soybeans or other ingredients in massive scales using these innovative kind of bioreactors. And I think that learning from that and thinking about how we can implement that in sort of waste conversion. And I actually think that if we could hook on an Ancham sort of production line onto a tofu factory, for example, Hodo Tofu in Oakland, the third largest tofu producer in the US, that's, I think, how we can get there. On site, add on to add value to the waste.
Awesome, thank you. That's a really exciting vision for sure. We had quite a few questions about how you're actually going about this fermentation. So in particular, you just mentioned solid state fermentation. So in your work, were you looking at solid state fermentation or submerged fermentation? How long does it take to grow, and what are kind of adjustments you'd need to make based on which approach you're taking?
Yeah, so Neurospora intermedia, it's fascinating. It actually is one of the faster growing edible fungi that we know of. So research was done in the 60s and 70s on Neurospora kind of for sustainable protein. And it's one of the faster growing fungi. It grows extremely rapidly. That's sort of its lifestyle. So, you can have in on-chum 36 to 48 hours, it's completed. Depending on the substrate, we might have to wait to 72 hours. That is a lot faster than many other edible fungi, including mushrooms that are very slow to grow and very slow to produce in the ways that we want. So in solid state, they grow very quickly. That is also true in liquid state. So one of the undergraduates, Alexandra Rosales, who I've been working with, have done really amazing work to test parameters for liquid scale fermentations, growing biomass on waste in liquid. And it's very quick. It grows on a lot of different things. And I think that ultimately it has applications in both kind of the solid state conversion, but also mycoprotein production, biomass for protein, as well as enzyme production in liquid state. So, I don't see it as, oh, this is only one solution. I think this has really opened doors to a lot of different directions and applications. And we're starting to understand the stress tolerance of this fungus, the pH range, the salt tolerance, to try to kind of optimize those and understand how we make the process predictable. Let's say we have a substrate and we know its molecular composition. Bam, we just have to alter these parameters to then be able to do a certain thing. That is sort of where we want to go, is to have it more predictable and more precise.
Yeah, kind of building off of that, I think one of the challenges that can emerge with using byproducts is that the feedstocks can be quite variable in terms of how often you're getting it, the quality of it, what you're getting. When you're thinking about scaling and when you're thinking about this distributed vision, how are you thinking about accounting for that?
I think that's where a lot of the fundamental research is going, is understanding Neurospora intermedia nutrient utilization. So again, with my talented undergraduate, Alexandra Rosales, we have started studying what are the nitrogen sources, what are the carbon sources that are being used across these different waste streams. And what then is the effect of alterations in those. And as I said, I think if we can, ultimately the vision is, and we're not there yet, but I think the research can get us there, is to say, let's first figure out what are the key things that Neurospora is needing. And then that we can do maybe a rapid test or just analyze the composition, let's say for the olive pomace harvest, for California in 2024, and say, okay, this is what it looks like. Now we know that these are the things we have to tune. Getting from substrate understanding to programming the fungal biology, that's kind of the missing gap that I think I'm really excited about addressing. And I think that will help us account for some of the variability. Now it's nice because Neurospora intermedia is already very robust. Clearly, across pHs, substrate type, and so forth, it will happily grow. And so I think we have a promising starting point to work with.
Amazing. It sounds like a really versatile organism.
I believe so.
We've talked a little bit about the scaling. We've talked a little bit about the costs. Can you speak to the regulatory landscape for these ingredients and kind of what it will look like on that side of adoption?
Yeah, I mean, I think one of the advantages of the Neurospora intermedia project that we did is there is a long-standing history of using this fungus, right? And we want to recognize and honor that history and make sure that, whatever applications moving forward is recognizing that, and also ensuring that we are not trying to profit in ways that are unethical of that history and cultural traditions. I think that's a more important kind of caveat to add, that we have to be sensitive about how we move forward. But I think for me, I kind of see a parallel to what has happened with koji or Aspergillus oryzae. I mean, some people on this call might be super familiar. It's like, this is like deep fermentation. If you follow Noma or some of these other fermentation establishments, I mean, Aspergillus oryzae is historically used in Japan to make soy sauce, sake, and miso, also used in East Asia more generally, Korea, China, Vietnam, other places, for various fermentation practices. And recently became a really powerful tool to transform plant-based ingredients into wonderful flavors in other contexts, be that Noma high-end restaurant in Copenhagen or home fermenters, koji has really domesticated us, and we really can see that it has so much promise and potential. So I think there's a nice kind of parallel there where there's a long history and we're continuing and building and innovating off that while also recognizing where it comes from. That also is promising for the regulatory landscape, because especially in the EU, when there is precedent prior to the 1970s, I believe, under the EFSA regulation of using the organism to make food, our path is much more clear in how this could be adopted. So I think that is like the historical context also gives us an entry point into that's how we can use it in restaurant, because it's considered, safe to eat, or it's considered, it's historically been used. That is going to be different when we look at GMO, when we look at synthetic biology. And so I think we have to engage with both approaches to have this impact, right? The understanding to manipulate whatever is there already, but then also the GMO to lead the frontier as far as what's possible. So does that make sense? I think sort of Absolutely, we can learn something from what's been done, and we can also take advantage of the fact that there has been something done for hundreds of years that suggests safety and promise, and then engage both GMO and non-GMO approaches in this space.
Yeah, it sounds like there are some really relevant parallels to draw from, so you wouldn't necessarily be starting from scratch, which is great to hear. Kind of on a different vein, how does this process impact allergenicity, and does it actually have the ability to reduce it at all for these food products?
I think there's so many outstanding questions that have emerged from this research, including this one. And that's the type of stuff that in my group we want to start investigating. It's like, okay, what is the precise nutritional impact, allergens, bioactives across all these substrates, and how do we then start optimizing for that? So I don't have the answer to you. We know from maybe work in sourdough fermentation that in the context of gluten, there might be kind of impact on gluten peptides if we ferment in certain ways. Open-ended question here, does it change the allergenicity of soy, for example, by breaking down those proteins or other molecules? Remains to be figured out.
Awesome, thank you. Yeah, there's also a question about whether there's any kind of metabolic crosstalk or nutrient exchange that occurs between the Neurospora and other microbes, and how that might impact food products that come out of it.
I think this is a great question, and it kind of gets to the fact that even though Neurospora is probably the main species that is driving on-chum production in Indonesia, there was other microbes present, and I think in that context, but also moving forward, the looking at and designing those community interactions is really intriguing for the robustness of the fermentation process, the kind of shelf life of the fermentation product potentially, and also the flavor. So I think there's a lot of room to both understand interactions, but also to really start designing and testing interactions. And I think this is one of the most promising directions moving forward, not only for this research in Neurospora, but generally in designing and making fermented foods. And my group will definitely start digging into this question. And I don't have the answer yet, but I think there's definitely elements there.
Absolutely. A lot of fascinating questions that I'm super excited to follow as you build up your group. You mentioned this study that you were able to do with these consumers, and you kind of just made a reference to flavor. So can you speak to some of the questions that you have about how this process is going to impact flavor, and potentially any flavor compounds that might be unlocked that are harder to achieve in other ways with these waste
materials? Well, so one thing we do know is that in on-chum traditionally, right, off flavors decrease. We see the production of, sort of earthy, mushroomy flavor. So aroma profile changes. We see the production of glutamate or basically umami. So free amino acids are being produced. We see the production of free sugars. We think that the free amino acids and free sugars can react to make the Maillard reaction happening, or browning, when we actually are cooking these foods. So, we have sort of a clue as to what might be happening. And I think the unanswered question is how, and what are the biochemical pathways? Like, how does hexanol decrease? What is the enzymes that are releasing all this umami and glutamate, right? So again, this is the starting point, and this is the awesome part, I think, of discovery work, that it opens all these avenues that we now are sort of uniquely positioned to pursue and interrogate. And I think in general, fermentation, in fermentation with fungi, we don't understand the link between a global change in flavor and aroma perception and biochemical processes. So there's a lot of room to figure that out too, to be like, well, what is actually going on and how do we find those enzymes? I'm really excited about this in the realm of mushrooms, especially, which have such a range of beautiful aromas and flavors.
Yeah, that would be an amazing link to be able to draw for sure. So again, I will look forward to learning from you as you dig into that. I also have a question about the process of doing research, because you have this beautiful vision of bringing together people from all these different disciplines. And one of the challenges that can arise is that the way that chefs do their prototyping and R&D, for instance, is potentially quite different from how a microbial engineer would do that, and everything in between. So as you're building out your lab and thinking about this vision and really creating this collaborative community, how are you kind of creating a common language and having people share methods effectively across that space?
I think as much as this work that's been done that I did has been about the research itself and the findings themselves, it's also about figuring out how do we work together, right? We have different incentives, we have different timelines, we have different cultures, right? And I think for me, having worked as a chef, having worked as a scientist, I feel that I've been able to speak both languages and also be sensitive to what those priorities and needs are. And I think moving forward, we'll keep figuring that out. And I think, some things that I've learned are, for chefs, for example, on the leading edge, sustainability is really important. If you can increase the economics and help the economics of your restaurant, that's awesome, right? An operation that wastes less and makes more, that's fantastic. There's also a very important element of creativity, right? So I think what this research opens up is all of a sudden you don't have to be using all the same strains and spores and approaches that everybody else also has access to on Amazon. You actually are able to pioneer kind of a new creative direction, opening up color, flavor, texture in totally new ways. And I think for us to appreciate that as a scientist, the things that we take for granted, oh, whatever, I can grow this, that might not seem that interesting to me because it's not hitting, the most fundamental process. It is incredibly powerful for opening doors and creative possibilities. So I think that the takeaway that I didn't share today is that I think we've learned a little bit how to work together. And for example, I've been at Blue Hill at Stone Barns 5 times over the last few years. And a lot of it's been, what conversations do we have? How do we work? And I think now we're locked in to be like, okay, here's what we see in it. And recognizing the need for the academic side, the rigor, going deep, that can take several years, but also then being able to move fast and prototype alongside that. And I think that one of the lessons here is that it's not only us bringing science to the chefs, but in prototyping and tasting, we are learning new science ourselves. When we tasted the rice when fermented with Neurospora and realized that it was sweet, we actually have uncovered, and I didn't talk about it today, a whole new set of enzymes used for plant biomass degradation, right? So there is room for learning on both sides, and I think that's awesome. And I think my space will hopefully continue to facilitate that.
Yeah, we have so much to learn from each other for sure. So it's great to hear that you've been able to build a system that works and continue to explore that. We have a question here about how you're thinking about producing meat analogs or new ingredients, or which categories you feel most excited about, and kind of like what feels most viable for the impact aspect of your work.
To be honest, I think this is something I've been thinking a lot about in the last few years. And I think that we have to make things that try to there's room in this space in general. We have to make things maybe that directly replace and mimic meat or other ingredients in a very direct way. I also think there's a lot of room, especially in getting chefs, to create foods that are just awesome, and they just are. Yeah. What is tofu? Tofu is not trying to be steak, it's tofu. What is anchovy? It's just anchovy. It's used in place of meat, but it doesn't look or taste like steak. And so I think for me, there's a really exciting frontier of leading the edge here to think about a realm of foods that provide the same satisfaction, nutrition as other things we're seeking to replace, but it's more about expanding and creating rather than trying to mimic one-to-one. Because I think in my research, and I think a lot of people out there have found, it can be extremely challenging. While there should be room for both, having worked with the chefs, I can really see the possibilities of just being like, this is an let's say a burger made from oat milk waste. Or fermented oats. Okay, and it's delicious. I want to buy it again. So let's kind of be sensitive to that too, and I'm very excited about pioneering that. And I think that's something we can do in academia, where we can be engaging with those more abstract directions.
Amazing. Thank you for sharing that. As a closing question for you, can you share a story about the most memorable prototype that you've gotten to work on in all of this, working with chefs, working with restaurants, working with different types of scientists?
Yeah, I think some of them are still, sort of like so exciting that I don't think I'm excited, ready to share it yet, because we're still sort of finalizing things. But I think, one of the things I think was what I shared with you today was really the moment where we brought Neurospora into Alchemist and we grew it. And some of the things were like, oh, this is bitter, this is dry. But the moment that we're all in the room together, tasting rice and recalling ripe fruit, bee pollen, flowers, it just, I think, for me, crystallized, okay, this is what this is about. It's about, we are just, through our amazing analytical instrument that is our mouth and our senses, we're able to kind of take things in directions that the technology alone maybe doesn't show us. And I think that kind of like fermented grain porridge dish, being able to transform rice and water and fungus, right, into something completely sweet, creamy, ripe fruit. I think that for me was probably one of the most memorable experiences I've had, and I think since it's just expanded, and hopefully in a few months or a year or so, I'll be able to share some of the really, I think, kick-ass prototypes that I think are poised to have a bigger impact, maybe.
Awesome. I think we're all eagerly awaiting that day. Thank you so much for this lovely talk and your really thorough answers, and best of luck as you set up your new lab. We're really excited to watch all of your progress and continue to learn about your research.
Well, thank you so much for hosting, and everybody for attending and asking these amazing questions, which I really enjoyed.
Awesome. Thanks, everyone. Thank you for attending, and we'll see you next month at our next Science of All Protein seminar.